So, Anamir, it's so great to have you here. Thank you for joining us.
Thank you so much for inviting me. Yeah.
So your company, Bexorg, takes real human brains that were donated after death, and you put it into this special system where you can study metabolism, you can kind of study how they function, and you can test some really important drugs that may address things like Alzheimer's and Parkinson's disease. So people have been doing human brain research for a long time where they got donated brains, they would slice them up, they would do things in the lab, but those are not active in any way. They're just kind of dead tissue. Right?
Yeah, it's a dead sort of like a static image of the brain. So in this case, this tool would require for cells to function and go through multiple of those slices.
So the dead brain just kind of sitting there, it's not going to send anything across. Right.
Exactly. And so to overcome this thing, there was this idea by Nenad, what if we can take a dead brain and do something that would allow us to deploy these tools and solve this big mystery of our brain, like how it's fired. And so we all got together at Yale, and we were like, we have to go to work now. And this really required, building, as of today, when we think about technology, what we have at Bexorg that stemmed from Yale, we had developed a device that can actually take these brains and provide fluid to these brains that can allow this mapping and software that can run the whole thing. So this all had to be developed.
But you need a brain.
You need a brain, yes. Yeah. We needed a lot of brains. We really had no clue what we were doing.
Whose idea was it to use pig brains?
So I think we were just thinking, I don't generally remember. I think it was more like we just need a lot of brains.
You need a lot of them. And they need to be big.
They need to be big.
Mammalian, large brains.
Large brain, yes.
So,
we ended up with pig, and luckily, if you think about, there's people like to eat pork, and people cull pigs, and so we were just going there, but no one eats brains.
Right. Okay, so you found a slaughterhouse in New Haven.
Yeah.
That processes pigs for food.
Yes.
Pork, all that stuff, right? And they don't use the head. They throw the heads away after they butcher.
Heads are not used, and these pigs are culled either way. So it was a really great opportunity for us to sort of get access to the tissue and also not sacrifice animals for research.
It's so interesting from a university standpoint, because I was on the ethics committee for animal research for many years at a medical school. And this category is really interesting because it's already, by the time the research team gets it, the pig's already dead and it's been processed for food. So it actually wouldn't have been qualified as animal research because you didn't kill the animal. Animal didn't arrive at Yale alive. You just got the brains from a facility that, again, processed them for food. So you're sort of it's almost like recycling. You're taking what would have gone thrown away in large numbers. And you said, well, we need large brains, we need large numbers to start your work. That was really an interesting approach, right? So it's not technically animal research because their animals are already dead. So you
it's tissue research.
Yeah, it's tissue research.
Absolutely.
Right. So tell me about the time you start getting the pig heads from the slaughterhouse, and how long did it take for you guys from after the pig was killed for food to when you got it in your lab and were able to work with it? What was the time period? So
the pigs would die, and from the point of death to when we would start our perfusion experiments, and this is the perfusion is a process in which we connect the blood vessels of the pig brain to our device and initiate our experiment. It was on average 4 hours.
So you have to get the brain out of the pig's skull. And there was no protocol or standard way to do that. You had to figure it out, right?
Yeah, so pig is pig is hands down the worst species that you would probably, in terms of anatomy, that you would
because the skull's super thick.
Yeah, it's super thick. They bump their heads. The brain is protected with a lot of bone. The vascular system is complex, to say at least, and so we had to do a lot of work on trying to understand how do we take the brain out, how do we connect the blood vessels, and just to get the experiment going. And this took a lot of iteration and failure. And I think with science, you really in engineering and science, you really have to be comfortable with failure.
So when you arrived from Croatia, you're at the Yale School of Medicine. You never imagined that you'd be sitting there with a pig head.
No,
trying to figure out
absolutely
how to
not.
get this brain out of
Imagine
here.
this, like you go, like I think to me it was one of those things, like you hear about Ivy League, you have ideas of like you have mental picture of what.
Right. Very sophisticated.
Yeah, exactly. You're going to the Department of Neuroscience at this prestigious university, but. Yeah, and I ended up like doing this, and I honestly, I think it was such a great experience, and I think it was amazing team that really understood like why this is being done and what's the end goal, and I think it required a lot of fortitude, and so you go from like, from these like harsh conditions, and you end up in the lab, and these were like long experiments that we would have to conduct. These would be like sleepless nights next to our machine that we were developing, and these fluids, and you have to make sure that you're capturing this biology. It was a really hard work, but I think amazing place, and I think really amazing environment to conduct
So you call
research.
you call this technology BrainEx.
Yeah.
X meaning like X outside the body.
Ex vivo, yeah.
Ex vivo.
Yeah.
So the pig brains were eventually taken out of the skull, and the carotid arteries were still intact. So you would hook them up to an artificial blood perfusion system, and then you published the results in the very prestigious journal Nature in 2019. What were the main findings that you were able to publicize in that article? Like what were the discoveries? Some of the main discoveries you had?
Yeah, so the main thing about that paper was discovery or realization that, once the heart stops and there's no blood flow in the brain, it's not really that like all cells just die instantly, which sort of we all taught the same way. I mean, I was in medical school like recently, and I was taught the same thing. So what we discovered is that this like process of cell death is a gradual decay. And that gradual decay can be actually exploited for science. So we can, take the brain out and put it into this environment that we created, and we can actually do experiments in this brain. So these are the two things. So the gradual decay of cell death, plus this retained experimental capacity that was within those brains. And so those are the two main things.
Yeah, what I found just so stunning about that work, and I ended up with a colleague writing one of the ethics commentaries for that Nature issue, was first thing that stood out to me was 4 hours after pig death, without any radical means to cool down the brain, nothing like extreme, just basically at room temperature, you were able to revive some basic physiological form after perfusion or during perfusion. You had some microcirculation of little tiny capillaries that are all throughout the brain. You put in a drug that for stroke patients will expand their blood vessels, and the pig brains responded to that in the same way. You can measure input of glucose and metabolism glucose, because everything you put in, you can measure coming out, right? Those are some remarkable things. So like basic cell function seemed to be restored. It completely shocked me because, as you said, I mean, I and many people thought as soon as somebody dies from lack of oxygen, right? So the pigs are slaughtered and they're bled out. So basically the brain dies from lack of oxygen. People assume that, your brain automatically starts to then just irreversibly lose everything that makes it special, all the functioning. Now, there were, as you know, there are accounts of people who have drowned in cold water and were like pronounced dead for a very long time, and they were able to be revived. And so there was maybe some idea of like, if you cool the brain, if you cool the body extremely low, then maybe you could revive them. But this is what was so stunning. It was room temperature. But was some of your work kind of at least informed by the idea that maybe there's something about the brain that's kind of more resilient than we thought after kind of lack of oxygen? Maybe from the
I think
studies?
Yeah, I think so. We also, we've seen these observations, as you mentioned, like someone falls in a cold lake and is fine after. There's, I think I mentioned before we spoke, there's this notion of if you are operating on a child that has a heart issue, back in the day, in some hospitals, even today, they stop the heart, they cool the kid down, the the newborn child to like 16C. They have like roughly around like half an hour to 40 minutes to operate on the brain, and then they're able on the heart, and the brain is fine, and they're able to restore the heart after. So, I mean, there were data points showing that this could potentially work. I think another thing is. People think I'm not sure that this would work well. The reason why, when we look at a human, let's say something happens to my heart and I drop here now, and it could be that you're not going to be able to resuscitate me, because there are issues with the blood as it starts to go through an injured brain and all those things. So by taking the brain out and putting it in our system, you're able to circumvent like some of the major issues that allow us to
that's a really interesting point. So the pig brains, and later very soon we're going to talk about the human brains. You take all the blood out of the brain before you put it into your system, and then the system has artificial blood, we'll just say, that goes through. That's different than real blood
in the human brain.
Yeah. And so you think that if it still had human blood in it, that the human brain in your system, it wouldn't work as well?
No, I think that the idea why everyone, including us also, to be clear, was kind of surprised with the discovery was because it is impossible to recreate this situation in a living animal. So it makes sense.
You have to have had the brain out.
Yeah, you need to do particular things, and we even say it in the paper. It has to be a particular set of actions that unlock this experimental capacity. Yeah.
So tell me then about the current work. So now you've said you've stopped doing. Bexorg has stopped putting pig brains into the system, and now you're using human brains. What do you do with those human brains in the system? Like, tell me about some of the things you're doing.
Yeah, we've been the reason why from day one we've been developing this technology in the first place is to enable experimental access in human brains. Okay. That's where the technology actually comes to its full potential. And the reason why we are doing it now, because we wanna address this one of the biggest unmet needs in the medical field, which is a lack of therapies for the brain. So
so brain disorders, brain diseases are not good therapies, and many people in the medical field realize that big problems are there aren't really good animal models to try to mimic or study human conditions of the brain, and so using real human brains is outside the context of a living patient. Is probably a pretty good middle step. Yeah.
So if you think about how we are developing therapies now, we start with model systems, so mice, organoids, cell cultures, and then we try to use those systems that are way simpler than the human brain. And the human brain is one of the most complex structures that we know of. And on top of the human brain, we also have a human disease, right? Which is another layer of complexity. So when you use mice or organoids or cell cultures, the first thing so there are a couple of assumptions or things that you have to do. You have to make sure that those models are representative of the human first, because your therapy is ultimately going into humans. And then on top of it, as you said, you have to develop a model of a particular disease in those models systems. And then finally, you go and start to develop your drugs or therapies, which are extremely difficult to develop. So to sort of like circumvent those issues, we have the ability now for the first time ever to take a human brain with, let's say, Alzheimer's disease or Parkinson's disease, because we can model those well, and then we can conduct these drug testing experiments
directly.
In these brains.
How many brains are you using at any, like let's say in a month? Like how many would you use?
It really depends on the study. So so one of the things that why we spun out the company from Yale, we wanted to industrialize this biotechnology that we developed and enabled it for scale and impact. And so that means that we now have the ability to take hundreds and hundreds of brains a year and really process multiple drugs or therapies that are
How long
potentially
does
going to
a
humans.
single individual brain stay on that system, and how long do you study it?
So when we start what we call the brain run, we keep it for 1 day, for 24 hours. So we standardize the protocol to 24 hours.
And you don't need any longer than 24 hours?
You do, of course, but it's the 24 hours is it's a viable protocol. It can provide answers that otherwise could not be like generated. And so, and we have done experimental runs where we extended the time on the platform. But it was more, if you think about the impact that you like to have on for patients and for these therapies to come out, this is a first tool of its kind. So we wanted to make sure that we can scale this thing and open it for society.
So walk me through kind of from sort of like the order of what happens. So from the time you get a brain to then you put it in the system, describe one study that you've done, and then what happens to the brain after. Kind of like walk me through
Absolutely.
that
So
course.
I think one thing is that a study is composed of multiple brains, right? This is simple biology. If you're running a biological experiment, you want to replicate experiments to gain like confidence in the results that you're seeing. So once the brain arrives to our facility, the brain is then placed in our system. And we initiate the process of perfusion, which means that we start to run this artificial fluid through the brain. This allows us to restore molecular activity in the brain. At that point, the system is stable. Then we take therapies, so they can be like small molecules, proteins, antibodies, like gene therapy, and then we can systemically deliver those therapies to the brain, and we can see how they go in the brain, where do they go, and how does the brain respond to these interventions or therapies, and we can collect all the tissue samples, fluid samples, and once the brain run is done, which is at 1 day mark, we store all those like samples, including the brain tissue, in our brain bank, and then we send it in the next couple of days for like data acquisition, as we say. So for genomics, transcriptomics, proteomics, metabolomics, we want to get like a deep molecular insights into what the human brain is doing.
Can you describe one exciting example of what you discovered using the system, kind of in your in one of your studies, like something that hadn't been discovered before, or people, maybe like a reason why a drug may have failed in the past, something?
Yeah, so we do a lot of what we call, as any other new technology, the big question is like, is this a good approach? Is the data we are generating, is it clinically predictive? And so the way, we go about answering this question is that we try to take like a clinical trial examples, outcomes that have been in humans and that are known, and so we call this reverse translation. So so we have done like two really cool examples. One is in the gene therapy space. So we deliver gene therapy through viruses, and so we can take a virus called AAV2. So in our case, we use it as a negative control because AAV2 should not go through the blood-brain barrier. It should not enter the human brain, while AAV9 should enter human brain. So those are known examples from the clinical trial. So then we take those same viruses, we deploy them systemically to our system, and they get delivered to the brain, and we can see actually that AAV2 does not enter the brain, while AAV9 does enter the brain. And so this is a really simple example of positive and negative control that provides validation for the platform that this is a viable approach to testing gene therapy before going to clinical trial. Another example is example of a failed drug called gamma secretase inhibitor. This is a drug that failed, I think like 12 or 13 years ago in the clinical trial.
So some other company developed the drug.
Yes.
lot of money, huge amount of time.
Yeah, absolutely.
They went to a clinical trial in living patients and it failed.
Yeah, it failed in the trial.
So why did you want to study that drug?
Because we wanted to see if we can replicate what happened in the clinical trial. Yeah, what went wrong. And so, because one of the things you want to, ideally, if a drug developer could do, is to know in advance if the drug is going to work or not. And we can do it now. Like, when we go to these like clinical trials, 95% of clinical trials in the brain, in the CNS space, they fail. If you look at, as I said, Alzheimer's or Parkinson's, we are looking at 99 to 100% failure rates in disease modifying therapies.
So you're saying before you even go to clinical trials with a bunch of patients, that you maybe would think that it's a good idea to run it through your system.
Absolutely. And then see if this drug is doing what it needs to do, or is the drug actually toxic maybe to the brain. And in this particular case, this drug had so this gamma secretase inhibitor had a toxicity issue in the clinical trial. And so when we ran the drug, so we took their clinical data, we ran the drug through our system and human brains that we had, and we showed that the drug was doing what it was supposed to do, which was always the case, also in the clinical trial. But we also detected signs of CNS toxicity. So if you're developing the drug now and you need to send it to a clinical trial, one would look at the data and be like, hey, I think we need to make the drug more specific. We need to go back to work before we put it into humans.
In that original human trial, were any of the subjects harmed during the trial from the drug?
I think, if I remember correctly, I think there were cancer instances and cognitive decline.
So what would be really great about the system that you have, if you did a preclinical study, it's interesting. It's a preclinical study, but it's using a human brain.
Exactly. So we're trying to bridge, like, I think the point that we are sort of saying is, like, let's take the mouse data out, and this is in broad strokes, let's take the mouse data out and do all the cool stuff that people in drug discovery are doing, but do it in these like molecular responsive human brains. And the idea is really simple. Like, we think that this is gonna bridge that gap when we go to phase 2 in particular, which kills almost all drugs in the CNS space.
So not only might it make it more efficient to develop more effective drugs, but also might make the clinical trials safer.
I think, absolutely. So if you look at the three main reasons why drugs fail, like for any drug in the CNS space, and like in particular,
CNS means central nervous system.
Central nervous system, in the brain. So you want to know, and we can't do like basic stuff because we can't open human beings while they are in the clinical trial. We can open up the skull, take a chunk of tissue. We just can't to see if the drug is doing what it needs to do. Is the drug even in the brain? So three questions that we try to address with our approach before going to clinical trial is what we call 3 pillars of drug discovery. So we want to measure exposure in the brain, we want to measure target engagement in the brain, and we want to observe functional pharmacology in the brain. So basically, confirm that the drug is really getting in the brain, confirm that the drug is hitting what it needs to hit and not other things, and see once it hits that thing that we presume that it should hit, it actually does what we presumed it would do. So these are things that we can address all like right now as of today using our system. And so that's what we are trying to increase the flow of medicine to like patients.
So I think a lot of people, and I'm sure you get this question all the time, will wonder, will these human brains ever wake up in the system? Will they ever regain consciousness? Because we were talking earlier about kids who drowned in cold water, and they actually were able to get revived, over an hour after they've been underwater. Is there a possibility of the brains coming back to awareness and consciousness in this system?
So I think the most direct and simplest answer is no, and the reason why is we don't restore neuronal firing, so there is no electrical activity in our brains. The way the system is designed, it doesn't allow for that to occur.
So you did things to try to make sure that wouldn't be possible?
Yeah, I think so. The technology is in place that just prevents this from like happening. This iteration of the technology cannot enable for neural firing to occur, disorganized electrical fire.
Why not? Why?
So there are a couple of things that we deploy in our perfusion protocol. So when these brains arrive, they're clearly isolated and it's just like a tissue, right? So there is nothing there. So once they are connected and we start to push this artificial fluid, blood through the brain, there are certain compounds inside that modulate how the cells work. And those compounds actually are preventing or not enabling, even providing the substrate for the neurons to fire. It's really expensive for a brain to fire. To us thinking here now and listening, that is energetically extremely expensive. So we never get to that state. And so that's one thing. And then there are different combinations of electrolytes and compounds that are sort of helping, not even helping, that are actually preventing from those things from
So it's
occurring.
not just oxygen pumping through that will wake it up. It needs a lot. You're saying electrolytes, all these other things that are not in your system.
Yeah. So there are things in the artificial blood or the fluid that we are pushing through the brain that is making, I'm trying to phrase this in the most accurate way, I'm not going into details, that's not allowing for cells to fire. And so they just don't have the substrate to fire. And we do confirm this. We have done what we call, it's called EEG. So it's basically like electric grid. So it's a recording grid that you put on brains and you can record if there's electricity in the brain. And
you did this with pig brains too,
We did this with pig brains. We did this with human brains. And we confirmed that there is nothing there. It's completely, there's no electrical firing, so zero, and that's our sort of like, these brains are by definition brain dead.
Is there anything you put into the Perfused just to make sure that they'll never wake up?
So we do a couple of things. We do, so it's a sort of like a layered approach. So we actually do, we remove substrates and modulate other substrates to make sure that it's impossible for cells to fire. And then as just in case, worst case scenario, and it's more like as a safeguard, we put also anesthesia, a particular compound that doesn't allow for cells to fire. To fire. So it's a layered approach.
So let's say the Alzheimer studies that you want to do, the Parkinson's, they don't need neuronal firing to give you meaningful results?
Absolutely. So I think the analogy I like to use with sort of what are we enabling with our technology in terms of biology and these human brains is, if you've ever seen Back to the Future film. So the DeLorean car.
The DeLorean car.
Yeah, the DeLorean car. So that's a car, right? It has like lights, engine, wheels. It's just a car. And so when Marty and Doc are driving the car around, they are driving a car. But once the car starts to speed up and it hits the speed of 88 miles per hour, it becomes a time machine, right? So that's basically what we discovered with all of our approach.
So the human brain's like the DeLorean, but it's not going 88 miles an hour.
Exactly.
You can do all the other stuff.
All other stuff. So it's not that the brain is not like a light switch, like 1 or 0. It's actually like a gradual thing, and that's the way to think about it. And so it's like a 0 and 100%. It's a dial. And so we are operating on the close to 0.
I see.
And this allows us to restore these like basic cell functionality. So, cells.
So you're doing your studies at kind of like low speed.
Exactly. So cells are making protein. Cells are removing waste. Cells are eating glucose and some oxygen. And so they're metabolizing. All these processes, what we call cell biology or cellular biology, are preserved and molecularly restored. And those same processes are actually affected in Alzheimer's, Parkinson's, FTD, ALS, and other neurodegeneration states of the brain.
So would you say that these that the brains in the Bexorg system, they're metabolically active.
Yes,
but
they are.
But they're not conscious.
They're not, yeah. So they're absolutely. It's like
so if it's like a dial, you're not getting it up to the level of activity where you would kind of have this like sudden like leap in kind of
not even
right?
close. Yeah,
And
like,
that's the consciousness. That's like the 88 miles an hour. What
it is absolutely like and to be completely direct here, I think we are like far from that. Like that's a hard technological and scientific feat, and where the platform and the technology is today, we are far from that point. Just
do you think anybody would want to try to take your system and try to do something like that though?
I'm sure that they would, yeah.
Do you control sort of the way the technology is used? Do you have kind of like special?
I think that's a great.
So somebody can't just like take your system and just do whatever they want.
No.
Because they need your permission, right?
So yeah, the technology. So that's a great question and the point. So technology is proprietary to Bexorg. We don't give out the technology, the devices, the artificial fluid, the software. That's that's Bexorg's. It's controlled, and that does not go out into the world. What does go out in the world are drug discovery efforts that that Bexorg does through either our own drug discovery efforts, or we partner up with reputable groups and people who are genuinely trying to develop therapies for the brain. But yeah, so it's all.
So speaking of the proprietary nature of the work you guys are doing, have people try to approach you and buy Bexorg and just kind of do other things with the technology? Have people try to?
I think it was yes. You can imagine 2019 like paper coming out. It made a huge splash. There was a lot of interest from different realms of society and people, but we were not interested in any of those things. And I think our goal from day one was thinking about like patients and
medical research.
Yeah.
And what were some of the things that people wanted to do with the technology?
Yeah. So you can kind of tier the whole approach. So there's clearly drug discovery effort or the medical research. Then there is the medical research through the through the technology in terms of like device or the artificial blood or software. So those are all like also like medical tools. And then the fourth one was organ preservation using this technology for kidneys or
other
lungs.
Organs that you can perfuse.
And then of course you can imagine the fourth
people who are like interested in eternal life.
Yeah, I don't think they gave it a lot of thought. Yeah, but it's fine.
Quite understood what you were doing.
Yeah,
that's amazing. So I want to ask you about sort of where you get the brains, how you get the brains. Tell me about that relationship you have with procurement agencies. How do you get brains?
Yeah, so all the brains that we use in our research are donations to Bexorg for research for drug discovery, which means that every brain that's a part of study for a particular therapeutic was authorized by the family, and they were informed about the research, and so they gave those brains to Bexorg to conduct our research. This is all done through organ procurement organizations. These are federal non-profit or organizations.
These organizations that get organs for like transplants.
Yeah, exactly. So they work. So they get organs for transplant, but also for research. Clearly, brain is not being transplanted, so it falls under tissue donations for research. But so we make sure when we work with them, and Bexorg basically uses same infrastructure that is in place that any other company or academic institution would use, and so that's how the process for.
Yeah, so normally I think the way it goes is if, let's say, hearts and kidneys are not high enough quality for transplantation, they could also be sent to research team to do research on these tissues. Clearly, the brain is not something that people are going to try to transplant into patients like a heart. And so it kind of falls under the research procurement group or arm, and that gets sent off to like research teams like you. So that's how you get the brains, and you get a lot of brains that way.
Yeah, so like these brains are, I think one thing that's really unique about our approach is that we discovered, that we can get brains from people that have been dead for hours. This gives us like a big window of opportunity, because we can take these brains and conduct research in these brains. Most people don't like to look at those brains because they don't have a technological tool that can make them useful. So these brains were otherwise, they would be buried and go to waste.
Even among brain researchers that use donated brains for research in their labs, not putting them to perfusion, but just slicing and looking at, like, these aren't even like the ones that they would really want because the time of death is too far.
Yes, yeah, and and that's the whole approach of the technology, and again, the punchline of the 2019 paper was that there is now a tool that allows us to take brains with this longer time and make them useful again.
But how long after death would you say typically brains are put into your system?
So we started with the pig, it was 4 hours, and now with the human, we are, we get brains that are up to 20 hours after death, and we are able to extract some useful information, some data points from these brains.
So these are donated by patients or their families to be used for research.
For research,
death.
yeah.
Do you ever get back in touch with any of the donor or the families and let them know what you learned from the research or how you use the brains?
So we are not doing that now, but we are. One thing that we are deeply looking into is trying to these community engagements and trying to understand what would people like to see, because we understand what we are doing, we understand how brain holds a special place in our hearts, and so I think so to be clear, we are not doing that now, but we are going out and trying to understand what can Bexorg do to provide something back to these families, besides eventually increasing the flow of medicine. But that's one thing, and some people would maybe like to know certain things, some would not like to know, but we had really instances where people who are familiar outside of the donation pool and people who are just close to Bexorg or are familiar with our research, and they had a loved one who was affected with Alzheimer's or Parkinson's, and we actually get phone calls and people
directly contact you.
Yeah, they wanted to give brains to us and maybe do something, because people are desperate for these therapies, and they understand that this is maybe a new way, a new opportunity that could provide those therapies for the next person that's gonna get affected in the family.
So if it's not really consciousness that's the main concern, what are some of the ethical concerns that you're thinking about these days?
Yeah, so I think there are multiple things here at play. So one thing, as you mentioned, is the consciousness and how we go about ensuring it is what it is, and there's really no suffering there. The other point is Bexorg is a commercial entity, right? It's a company, it's a business. So it's this idea of giving back to the community and how we are thinking about what's the give back. And so, one thing is high level. There's a idea that this enablement through technology can increase the flow of medicine to the generic space, aka that every human being can benefit from these new therapies.
Yeah. So you are enabling the discovery of better, more drugs, and that can become more available to
exactly. So because every drug eventually will become a generic drug, implying that penicillin maybe not the best because it's antibiotic, but let's think about Advil, right, it's invented, it does its job, it's still going to work in 500 years. So this idea of we are providing more tools for every human being to be treated with. And then the other thing is maybe more direct give back. What's Bexorg doing to give back to the community right now, short, mid-term?
So people who've donated donated brains?
So maybe not. These are, and we have our ethics advisory board, and these are the questions that we are trying to understand and discuss with the ethics advisory board through the community engagement as well. What are the right ways for Bexorg to give back to the community? So, we can work with patients, as we do now. We work with patient advocacy groups. We can maybe work with patient caregivers. We can maybe do something directly for the family of those that have donated the brains for research. So there are a lot of things that Bexorg can do. We had one on one of the recent engagements, which was really nice. And this was idea from the community. They said, maybe we could also create basically a center or a place where people who decide it's fine to say look, someone from the family gave the brain, or the person consented, maybe they would like to show look, IZ gave the brain to Bexorg, and look what I did. Sort of because people are genuinely doing this because they want to help, and then showcase, putting a face to it might help. So there are a bunch of things that can be done, and we are now looking into this and trying to understand what's the best approach. And I think it has to be done through a conversation with the community, and that's what we're doing now.
That's interesting. So the ethical issues that are top of mind for you is how can Bexorg be a more socially responsible company?
Absolutely. Look, I think we are. I think of us as we are getting these gifts of donations. That enables our research. We are stewards of the tissue, which means that we have to behave in a particular and respectful way. And then it's also we are building something new together, and there's going to be a clear benefit if everything works out. So how can we also give back to the community? And that's what we are trying to do now. Understand, we are engaging, as I said, ethics advisory board, community, to try to understand what's a reasonable and a nice way of giving back, because it's all about the future success, and it's really, there's nothing, it's just upside. So upside can be shared, and it's really something that we think about a lot, and we are now trying to understand actively what are the best ways to achieve that.
If there's a researcher who's trying to develop a drug for, let's say, earlier stage Alzheimer's, so not late stage, earlier stage, is there a way that you could find potential donors who maybe they might agree they're recently diagnosed with Alzheimer's and maybe they die of other causes? Could you get brains that are more early stage disease, or are they all basically going to be very late stage?
So because the way this is done, we get brains that are different stage of the disease development, so we can then map out how the disease progresses. We don't know in advance if a particular brain is coming, where exactly it falls on the disease progression. But
you do get a range.
You do. And because these people die also from other things, and it might happen that you might have a clinically healthy human that passed away, or a person, and we might get that brain for research, and then we are going to conduct a particular experiment, and then subsequently when we do, or when we look into that brain, we might uncover that it's not just that Z was forgetting things, but Z was actually at the early stage of Alzheimer's, and then we can reclassify that brain even more accurately than.
Do you ever get any football players that want to donate their brains for about CTE or something that they care about?
Well, we did not. I've been getting that question a lot.
It's football season now.
Yeah, it's football season. Yeah. No, we have not done. So we've been solely focusing on Alzheimer's and Parkinson's, and just broadly neurodegeneration. But this is one of the big things that keep coming up. Yeah,
because I have heard a lot of football players, they have sort of in their wills or something, like to donate their brains to science after death, because they very much care about in the professional sport all this happening with CTE.
Yeah.
It's really interesting. I didn't talk with you about the Perfused that you use, but that's something that was invented at Bexorg. So the artificial blood that kind of goes into the system. It's not real blood. Tell me about that perfusion.
Yeah. So just maybe high level, there are three core pieces to technology, or four now. It's the device. There is the artificial blood, which you are referring to, or the perfusate. There is the software that runs the whole thing, and now there is we have like AI models now being derived from the data points.
You use AI in your system?
Yeah. So in terms of the perfusate, we use, compared to maybe other perfusion groups that are out there, we use cell-free approach, which was also in 2019 in the paper. So we don't use real blood to push through the organ.
Why don't you use real blood?
I don't think it scales well.
So you can't get enough.
You can't get enough
in the quantity that you need without kind of harming the blood supply for
yeah, I think
patients.
it's. I mean, the first paper was a nightmare to get out, and so there were a lot of experiments that had to be done. So imagine now you're going through the iteration phase and you're just wasting blood, not like continuously. That's not scale. That does not scale. So when you think about technology, it's not even like the coolest, the sexiest tech is going to be when it's the one that scales and that's impactful.
Yeah, so by scale, you mean like you can generate huge quantities.
Yeah, because you have so many questions. Like these are complex environments, complex devices, complex like protocols. You have so many questions that you have to ask and address and fine tune, and then you want to industrialize the whole thing after. You need scale, you need numbers. So using blood was from day one out of the question, and I think it was a really challenging effort trying to build like a fluid that is blood-like, and we're far from it. Trust me, like it is.
It's pink, right?
It's kind of like red. Yeah, it has like a red color to it, and so we use cell-free approach. We make it completely in-house now.
So it's cell-free. How does it carry oxygen?
So it has a particular compound that we extract from a worm.
From an earthworm.
Yeah, and people have done this before. We just sort of like put everything together in a particular form. So we extract it, and it's a potent molecule.
So it's a little molecule, but it can carry oxygen, like a red blood cell would be.
Exactly, yeah. So
it's much smaller.
Much smaller. So the red blood cell, like a human red blood cell, is around like 5 microns in diameter. This molecule is like a small ball, maybe like 20 to 30 nanometers. So it's
way smaller.
Couple of it's one scale below. So it's really small, and this allows us to develop like a scalable solution that can then get throughout the whole brain, can go around stuff, and get into all places.
So it's the system, the Perfused, can you kind of like recharge it and can reuse it again, or is it just like this is just one-time use?
So it's one-time use. So one brain gets one sort of like fluid, because that fluid then becomes enriched with the information from the brain. So, and we actually store that fluid. So once our brains, even as we speak now, are taking molecules from our blood, right? And our brains are releasing new molecules into our blood and into our CSF, which is the fluid in which the brain is like it kind of like floats within this like CSF fluid. So as these molecules are being released, those molecules are also being released in our system, and so this artificial fluid that we put in, and it captures all that information. We actually use that for drug discovery, so we can know, is brain actually responding to a therapy? How is it responding? Do we need more or less of a particular intervention, and so on?
And then the Perfused that you folks developed, could that be used clinically in other ways, other contexts?
I don't think in this iteration, no. I think it would have to be like a procedural, like there's a long way to go to. I think it's directionally could be used, but to get it to a practical point, it needs a lot more work. Yeah.
Yeah. What were, besides people wondering and asking you quite a bit about consciousness coming back in the brains, and we addressed that already, what are some other misconceptions that people have had and have said about, the work you do that you would like to respond to?
Yeah, I think, well, the one that you just said was this idea of like, is there something there? Which there isn't. These brains have no EEG, there's no electrical firing, and we have validated these protocols and we have shown this. I think another misconception that people have, and we briefly touched upon this, is this idea that either brains are absolutely doing 100% of the work or nothing. Which is something if you're trying to no, that's not the case. It's actually the brains can be like slightly molecularly active, and it can be exploited for science and drug discovery. I think there's also, because of the novelty of the work, and people are when we people don't understand that these are gifts these donations are gifts to Bexorg, to science, to the community. So I think the way of how we go about treating those gifts in terms of how we talk about them, how are we showing the ultimate respect to every single brain that was given to Bexorg or to any science group. So just the idea that there needs to be a particular kind of stewardship and ownership over the tissue, and there needs to be like a responsibility and the accountability. And we do all these steps.
So you take it very seriously.
Absolutely.
Showing respect for the families, for the donors, for the tissue that you're using, for the brains. And so it must be kind of annoying then to hear people say things, I don't know, like just clickbait or whatever. People just trying to raise rage.
Look, it's novel research. It captures imagination, and so when someone goes to talk about this, they talk about brains in buckets, or or they say these brains are alive or something. We never use that language. These brains are not alive. They have viable cells that are molecularly responsive. These are not living brains. These are not brains in buckets. These are medical chambers that are high tech. They have sensors, and they're trying to emulate, some physiological environment. So, they're actually like, if you genuinely open up the system and you look at it, you would see it's kind of like boring. In a sense, it's just like electronics and science and some data points. But there's this like language that gets associated with these brains, and it's and at the same time you have these gifts, that someone gave. So you have to kind of, so we try to go out and say like, hey, like, look, like we should be respectful. And because eventually, there is going to be something, some drugs will come out of this, and someone, these brains will help us or enable us to like put these therapies out. So that's like a big challenge. I think it's also like a question of time, because it's a new thing, and so it's just going to take a bit of time for people to get like comfortable and with the language, and learn like what's sort of okay and what's maybe not okay. Yeah.
So explain to me, or describe to me, the first time you used the human brain in the system. That first one for you. What was that like? Well,
I think for me it was I think it was 6 or 7 years in the making. And because, as I said, we were developing this in the pig brain. We worked with pigs. And, finally the day came when we got the first human brain. And it was one of those things where like you spend like years and years of research, like to a particular point, it culminates with a single first brain. And I just had like a set of flashbacks, from like starting with the pig, now seeing it seeing the human.
You see
brain.
like that video montage, like Croatia, Yale.
Yeah, everything is coming. You see it in my brain. But no, it was really like it was a big thing, and we were all excited because it's finally it's happening. And I think maybe beyond the first human brain, I think what was amazing was what took us 6.5 years, like roughly, to achieve with the pig. We achieved with just 6 human brains, and that's the value of the pig as a model that we use to like learn to develop the platform, to learn how to get these data points, to learn how to use the tissue. Like we were able to achieve all those things, like compress it within like 6 to 7 brains, and I think it was really amazing. And then just start
Where do you think the future of this work is going? In the next 10 years, 5 years, where do you hope, or where do you see it going?
I see that the therapies are going to come out for patients that were developed or have been developed like using our approach and our technology, our platform. So it's all about like, we're all doing this because the only thing we want to see is these like therapeutics, like, or medicine coming out and treating Alzheimer's, Parkinson's. That's the only thing we care about. And I hope that we turn this like 99, like 95, like 99% like failure rate that we have in the CNS space. We turn that into a success rate. So we would like to see the technology like validated and disseminated throughout the whole industry and academia, so people can like really do new amazing neuroscience that wasn't possible before, but also put therapies out. You look at the whole sort of work that Bexorg is doing, and what's being done at Bexorg and what was done at Yale, and it is cool science. It is if you're a neuroscientist, it is the model, which is the human brain, and you want to learn. We all want to know how it works, and we want to do research in the human brain. But I think the main point to really understand is that this is the reason why this is being done, and this is being done not because it's cool science. It just happens to be really amazing, and I want like some kids somewhere to listen to this thing and be like, hey, if this guy from Croatia can go and do it, then I can do it too. But it's also about like a hope for patients that have not seen these therapies, that like they're just not out there. So this is why we are doing it. We are doing this because we want to put therapies out, and for the first time ever, we now gain experimental access to like real human biology, and we want to find like through ourselves and partners, we want to impact patients as quickly as possible. So that's why we are doing it, and would love to have everyone who wants to do a deep dive can come, and like we're really transparent about our work and open, and would really like to make it like a gold standard for the whole industry. And that's maybe the main thing to take away.
Well, thank you for all the amazing work that you do and the incredible innovations that are coming out of your team. Thank you for joining us today.
Thank you so much.